morning guys. In this video we will see about the next water vitamin B12. So like any other vitamin, this also I will be discussing under my usual headings. To start with, a brief introduction, then RDA and sources. When you consume these sources, how they enter into our body. After entering into our body, what are the functions it executes? If these functions are not done, what are the deficiency manifestations it will cause? And if these functions are overdone, what are the toxic manifestations you will get? Okay, so these are the headings under which I will be discussing about vitamin B12. First, we will start with a brief introduction. So this vitamin B12, the structure of vitamin B12 is almost similar to that of heme. Okay, we all know that heme is made up of porphyrin ring, but our B12 is made up of a similar ring called as chorin ring. This is also like a porphyrin ring only. It is also made up of a tetrapyrrole structure, but in heme, there at the center we will be having an iron atom, but here instead of iron we will be having cobalt atom. That is the difference. Okay, so this chorin ring is also a tetrapyrrole ring structure with cobalt atom at its center. Okay, so now the valency of this cobalt is 6. Out of this 6 valency, the 4 valencies will be bound to the 4 pyrrole structures. Okay, the tetrapyrrole ring, the 4 pyrrole rings, it will be attached to the cobalt. Okay, now to the 5th valency, if you attach a Benz imidazole ring, then this chorin ring will become cobalamin. Okay, to the 5th valency, if you attach a Benz imidazole ring, then chorin ring will be converted to cobalamin. Okay, now to the last valency, 6th valency, if you attach a cyanide group, then it becomes cyanocobalamin. Okay, if you attach an hydroxyl group, it becomes hydroxycobalamin. Okay, instead if you attach an adenosyl group, it becomes adenosylcobalamin, or shortly called as ADO-B12. Instead, if you attach a methyl group, then it becomes methyl B12. Okay, so these are the various forms of cobalamins. Okay, cyanocobalamin, hydroxycobalamin, adenosylcobalamin, and methylcobalamin. Okay, among these, the cyanocobalamin is the one which is predominantly found in the oral supplementation, especially in B12 tablets. Okay, all the B12 tablets you are taking, they will contain B12 only in this form, cyanocobalamin. Whereas all the injectable forms, vitamin B12 injections, okay, so they will be having this hydroxylcobalamin. They will be having this hydroxylcobalamin. Okay, so both the cyano or hydroxyl, after entering into our body, it will be converted to either to adenosylcobalamin, okay, which is actually the storage form of B12, or otherwise it will be converted to methylcobalamin, which is the circulatory form of vitamin B12. Okay, so among all these forms, the storage form, adenosyl B12 and methyl B12, they have certain biochemical functions in our body. Okay, so these two forms, they are considered as active forms of vitamin B12. Okay, so this is an introduction about B12. Okay, so we have different forms, cyanocobalamin, hydroxycobalamin, adenosylcobalamin, or methylcobalamin, depending on to which the 6th valency of cobalt is linked. Okay, if a cyanide group is attached, it is cyanocobalamin. Okay, if an hydroxyl group is attached, it is hydroxycobalamin. If an adenosyl group is attached, it is called as ADO-B12. If a methyl group is attached, methyl B12. Okay, in this adenosyl B12 is the storage form, methyl B12 is the circulatory form, and these two has a important biochemical function in our body, which we will be discussing later. So these two forms are called as active forms. Okay, so next coming to the RDA, recommended dietary elements. Among the vitamins, vitamin B12 is the one which is needed in very least quantity. The recommended dietary elements for vitamin B12, it is just 1 to 2 micrograms per day. We need only 1 to 2 micrograms per day. So this B12, from where you can get it means, you can get it mainly from the animal sources. You can get it mainly from the animal sources. Okay, vitamin B12 actually it is a quite unique water soluble vitamin. Okay, so how it is unique means it is unique, it is different from the other water soluble vitamins by two different properties. Okay, so first one is usually all the water soluble vitamins, they will not be stored in our body. The excess will be excreted from our body through urine. But vitamin B12, it like fat soluble vitamin, it can be stored within the liver. Okay, so this liver, animal liver, acts as the richest source of vitamin B12. Okay, so that is the thing. So animal liver is the richest source of vitamin B12. Apart from that, it is also present in other animal sources like meat, egg, all these things it is there. Okay, but it is completely absent in plant sources. Okay, vitamin B12 is present exclusively in non-veg food items only. Okay, if that is the case, what will happen to the vegetarians? How they will get B12? They can get B12 only through curd. Okay, even in curd, actually in curd we will be having lactobacillus. So that lactobacillus has the capacity to synthesize vitamin B12. So that is the only source of vitamin B12 for vegetarian people. Okay, so that's the reason vitamin B12 deficiency is more common in vegetarians. Okay, apart from this, even the bacteria present in the intestinal flora, okay, they can also synthesize vitamin B12 and they can also add up to the daily requirement to some extent. Okay, so these are the three different sources for B12. Okay, it is rich in animal sources and vegetarians can get it only from curd or from the intestinal flora. The bacteria in the intestine can also synthesize to some extent. Okay, so now when you consume all these things, how it is absorbed in our body, we will discuss next. Initially I said there are two important differences between B12 and other water soluble vitamin. One is like fat soluble vitamin, it can be stored in the liver. Second thing is like fat soluble vitamin, here also for the absorption of B12 we need lot of proteins and the absorption is quite complex. Okay, so how it is absorbed, the dietary B12, how it is absorbed into our body, we will discuss in detail now. Okay, so in the diet our B12 is always found in a bound form. They will be found bound to protein in all the animal sources. When you consume this, what will happen means it will be entering into our stomach. In the stomach, by the acidic pH, these proteins will be digested and free B12 will be released. Okay, in the stomach, because of the gastric pH, the bound form will be converted to free form. Now, this free B12 will bind to another protein called as cobalamin. Cobalamin, which is produced from salivary gland, Okay, so when you consume these sources along with this food, when it enters into the mouth, this saliva will be produced. In saliva, this cobalamin will be coming. So all these together will be reaching the stomach. So in the stomach, this protein will be digested. Okay, whereas this cobalamin is acid resistant. Okay, so they will once this is digested and B12 becomes free, it will go and bind to cobalamin, and in this bound form they will reach the intestine. So once it reaches the intestine, the pancreatic enzyme trypsin, they will digest this cobalamin and again they will release the free B12, free vitamin B12. Now in the intestine, once the B12 is released, it will bind to another protein called as intrinsic factor. Okay, so this intrinsic factor is a protein which is produced by the parietal cells of stomach. Okay, so from stomach it will be coming out. So once it reaches the intestine, when trypsin digests cobalamin and releases B12, our intrinsic factor released from parietal cells of stomach will go and bind to the B12. Okay, so once it binds, the entire complex will be absorbed and it will be entering into the body, and the absorption takes place at ileum. Okay, the absorption of folic acid takes place in jejunum, whereas the absorption of B12 along with the intrinsic factor takes place within the ileum. Okay, so after reaching the after entering into our body, after entering into the circulation through ileum, it will enter into the circulation. Okay, so in the circulation, the B12 will be again released. Okay, and it will be bound to another transport protein called as transcobalamin. Okay, specifically it is trans-cobalamin 2. Okay, so the B12 which is entering will bind to trans-cobalamin in the circulation. Okay, now this trans-cobalamin will take the B12, okay, and it will be storing it in the liver as, what is the storage form I said initially, as adenosylmethionine B12 or otherwise called as ADO B12. Okay, so this is how B12 from the diet is absorbed, enters into our body, and it will be stored in the liver. Okay, so how much we can store means human liver can store up to 200 milligrams of B12. Okay, the normal dietary requirement, daily requirement is just 1 to 2 microgram. Okay, that means liver can store up to 1000 times the requirement. Okay, so the deficiency of vitamin B12 is quite rare. Usually, even if you don't take vitamin B12 for 2 to 3 years, there won't be any deficiency. Only after that the manifestations will start appearing, because the liver has that much good storage reserve. Okay, so that's it about absorption. Okay, so for absorption we need many things. What are the things? First of all, gastric pH, acidic pH is very important. Only then the cobalamin will be digested and B12 will be released. Okay, if this is acid pH is not there, what will happen? Cobalamin, it will not be released from the free form, it cannot bind to intrinsic factor, and it cannot enter into the circulation, it cannot be absorbed. Okay, so gastric pH is the first thing which we need. Second, we need trypsin, okay, which is a pancreatic enzyme and digests the protein cobalamin. Okay, so to digest that we need the pancreatic enzyme trypsin. Okay, so we need gastric pH, we need trypsin, and finally we also need intrinsic factor. Okay, so all these three things are very much important for absorption of our vitamin B12. Okay, so after absorption, after entering into our body, it will be stored in the liver, and whenever it is needed, it will be released from the liver, and they will be doing the biochemical functions. Okay, so what are those functions? We will discuss next. Now we will discuss about the biochemical functions of vitamin B12. So initially I said that there are two active forms of vitamin B12. One is adenosylmethionine B12, or otherwise shortly called as ADO B12, and the other one is methyl B12. These are the two active forms. So each of these active form has one important function in our body. First we will see about this adenosylmethionine B12. So what is the function, biochemical role of this adenosylmethionine B12? Means it is very much needed for the conversion of propionyl coenzyme A to succinyl coenzyme A. Our vitamin B12, adenosylmethionine B12 especially, is needed for the conversion of propionyl CoA to succinyl CoA. Okay, so we all know that propionyl CoA is a 3-carbon compound, whereas succinyl CoA is a 4-carbon compound. Okay, so how to convert a 3-carbon to a 4-carbon compound? By adding one more carbon. Okay, so this addition of carbon is called as carboxylation reaction, and for any carboxylation reaction the enzyme is carboxylase and the coenzyme is biotin and ATP. Okay, so our enzyme propionyl CoA carboxylase, it will add one carbon atom. When it adds one carbon atom, it will not form succinyl CoA, instead it forms methylmalonyl CoA, which is actually an isomer of our succinyl CoA. Okay, when you add one carbon, it is converted to methylmalonyl CoA, which is an isomer for succinyl CoA. Okay, so now how to convert this methylmalonyl CoA to succinyl CoA? I said they both are isomers. Okay, so by using an enzyme isomerase, they can be interconverted. Okay, but the enzyme isomerase, methylmalonyl CoA isomerase, it will act only on L form, not on D form. So first, by an enzyme called as racemase, this D form is converted into L form, L-methylmalonyl CoA, and then the isomerase, the enzyme methylmalonyl CoA isomerase, can be used for converting it into succinyl CoA. Okay, so this is how normally propionyl CoA is converted to succinyl CoA. Here, what is the role of our B12? Means our adenosylmethionine B12, it acts as a coenzyme for the enzyme methylmalonyl CoA isomerase. Methylmalonyl CoA isomerase. Okay, so thus it helps in the conversion of propionyl CoA to succinyl CoA. Normally, this propionyl CoA is formed in our body during odd chain fatty acid catabolism, or during the catabolism of valine, isoleucine and methionine. Okay, so during the catabolism of these components, okay, odd chain fatty acids, valine, isoleucine and methionine, during their catabolism, propionyl CoA will be formed. Okay, that propionyl CoA will be converted to succinyl CoA with the help of our B12, and this succinyl CoA can either be oxidized completely in TCA cycle to produce energy, or it can be utilized for glucose synthesis through gluconeogenesis. Okay, so ideally, so finally, what is the ultimate function of B12 means? It helps in the catabolism of these compounds, odd chain fatty acids, valine, isoleucine and methionine, and helps it to get converted into glucose. That is the function of adenosyl B12. Okay, so function of adenosyl B12 is it acts as a coenzyme for the enzyme methylmalonyl CoA isomerase. By acting as a coenzyme, it helps in the conversion of propionyl CoA to succinyl CoA. Okay, so how propionyl CoA is formed? During the catabolism of odd chain fatty acids, valine, isoleucine and methionine. Okay, so these compounds, it will be converted to glucose. It will be utilized for gluconeogenesis only in the presence of adenosyl B12. Okay, fine. So next, coming to methyl B12. So this methyl B12, what is its exact role means? Like here also, like adenosyl B12, this also has only one function in our body. So what is that function means? It already in folic acid metabolism, we saw that methyl folate, okay, methyl tetrahydrofolate is the one which donates the methyl group for homocysteine to methionine conversion. Okay, for the conversion of homocysteine to methionine, the methyl group was donated by methyl tetrahydrofolate. This we already discussed under the folic acid metabolism. Okay, but this methyl tetrahydrofolate, it cannot directly give its methyl group for this reaction. Instead, what they will do means, first they will give it to the B12, vitamin B12. Okay, so it will give it to the B12 and converts this B12 into, so this methyl tetrahydrofolate, it will be donating the methyl group to the B12 and it will be forming methyl B12, releasing the free tetrahydrofolate. Okay, so now this methyl B12 will be donating the methyl group for this reaction and helps in the synthesis of methionine. Okay, so the enzyme for this reaction is nothing but methionine synthase or homocysteine methyltransferase. Okay, so for this enzyme, our B12, methyl B12 acts as a coenzyme. Okay, so this is the function of methyl B12. Now, by receiving a methyl group from methyl tetrahydrofolate, what it does, it relieves the folate trap. Okay, normally in our body, tetrahydrofolate has a tendency to get trapped as a methyl tetrahydrofolate. Okay, if it gets trapped as methyl tetrahydrofolate, it cannot be used for other purposes. Okay, that is prevented and free tetrahydrofolate is released. Okay, folate trap is relieved and free tetrahydrofolate is released by our vitamin B12. So through this reaction, it helps in relieving the folate trap. Okay, that is one function. Second, by donating the methyl group to homocysteine, it results in the formation of methionine. That methionine in turn can be used for various transmethylation reactions. Okay, so actually methyl B12, it acts as a coenzyme for only one enzyme, homocysteine methyltransferase, or otherwise called as methionine synthase. By acting as a coenzyme, it does two works. What are the two works? One is it relieves folate trap, and second one, it helps in transmethylation reactions. Okay, so through this transmethylation we get lot of products. For example, choline, okay, which is needed for the myelination process, is synthesized by this transmethylation reaction. Then various catecholamines, okay, for example, norepinephrine is synthesized. Okay, then even melatonin is synthesized. Okay, so for all these things we need transmethylation reactions under our vitamin B12. That's it about the functions of vitamin B12. To sum up, the two active forms of vitamin B12 are adenosyl B12 and methyl B12. Okay, so each of these acts as a coenzyme for one particular enzyme. Adenosyl B12, it acts as a coenzyme for the enzyme methylmalonyl CoA isomerase. Okay, by acting as a coenzyme for this, it helps in the catabolism of odd chain fatty acids, valine, isoleucine and methionine, all of which are gluconeogenesis substrates. Okay. Next, coming to methyl B12, it acts as a coenzyme for the enzyme homocysteine methyltransferase or methionine synthase. Okay. By acting as a coenzyme for this enzyme, it helps in relieving folate trap, and also it helps in transmethylation reactions. Okay. So that's it about the functions of vitamin B12. Next, we will move on to the deficiency. So regarding deficiency, you should always mention three things. One is causes for the deficiency, second the deficiency manifestations, and third how to diagnose it. Okay. So first we will start with the causes for vitamin B12 deficiency. So this vitamin B12 deficiency can happen because of two reasons. One is decreased input or otherwise intake, decreased intake, or increased output, or otherwise we can call it as excess demand. Okay. So if any of these things can lead to vitamin B12 deficiency. First we will start with the increased demand. Just now I said that vitamin B12 helps in relieving folate trap. Okay. So for the proper function of folic acid, vitamin B12 is very much needed. Okay. So what is the main function of folic acid? It helps in purine and pyrimidine synthesis, which in turn is needed for DNA synthesis, which in turn is needed for cell division. So whenever there is increased cell division or whenever there is an increased cell turnover, parallelly the demand for folic acid as well as the demand for B12 increases. So during pregnancy, then in case of any hemolytic anemia where there is excess cell turnover, and also during cancer. Okay. So where there is increased cell division. Okay. So all these three conditions, they will increase the demand and it leads to vitamin B12 deficiency. Next, coming to decreased intake. So this decreased intake may be due to two things. One is dietary deficiency, when you are not taking good enough quantity of B12 in your diet. Okay. So that is more commonly seen in vegetarians. Okay. Vegetarians are more prone for vitamin B12 deficiency because the most of the B12 is present mainly in animal sources. Okay. So decreased intake means it can be either decreased, it can be due to dietary deficiency, or on other hand it can be due to decreased absorption. Okay. You may take enough B12, but if it is not absorbed, then also it can lead to vitamin B12 deficiency. Okay. So this decreased absorption can be either general or specific. It can be either general or specific. Okay. So general means there are few general conditions like tropical sprue, celiac disease, or any gastric surgeries. Okay. In all these conditions, what will happen? Digestion and absorption of almost all the nutrients will be affected. Okay. So along with that, you will also be having vitamin B12 deficiency. Okay. They are called as general malabsorption syndromes. Coming to the specific part, we know that the absorption of B12 is quite complex and it involves lot of proteins and other requirements are needed. Okay. So when those things are affected, it specifically affects the vitamin B12 absorption. Okay. So what are those conditions? Means, to start with, we need gastric pH, okay, acidic pH to release the dietary forms, okay, from the bound protein. To release the B12, we need acidic pH. Okay. So whenever the gastric atrophy is there, which cannot produce a proper HCl, or if you are taking proton pump inhibitors, antacids, okay, proton pump inhibitors in excess quantities, okay, or if there is a condition called as achlorhydria, where the acid production by the gastric cells are defective. Okay. In all these conditions, your vitamin B12 absorption will be affected. Next, second, we know that we need also trypsin, okay, which actually digests the protein cobalamin in the intestine and releases the B12. Only then it can bind with the intrinsic factor. Okay. So whenever there is a pancreatic disorder, okay, pancreatic insufficiency, and there is decreased production of pancreatic enzymes like trypsin, that can also lead to vitamin B12 deficiency. Okay. Next, the third and the most important requirement for B12 absorption is intrinsic factor. Okay. So when that intrinsic factor is deficient, that can also lead to anemia. Okay. So there is a specific condition called as Addisonian pernicious anemia. Addisonian pernicious anemia, okay, which is nothing but a autoimmune disorder where antibodies are produced against our intrinsic factor. So what will happen? This intrinsic factor will be damaged. There won't be any intrinsic factor available. So vitamin B12 absorption will be affected and it leads to anemia. Okay, so that condition is called as Addisonian pernicious anemia. Okay, so here also B12 absorption will be grossly affected. Apart from that, drugs like metformin, okay, in diabetic patient it is more common. Okay, metformin is one of the commonly used first line anti-diabetic drug, okay, oral hypoglycemic drug. Okay, so when you take this metformin, it can affect the B12 absorption and they are more prone for vitamin B12 deficiency. And finally, consumption of fish tapeworm. Okay, there is a particular worm infestation called as fish tapeworm. So when you are infected by that can also affect the vitamin B12 absorption. Okay, so these are some of the causes where the patients may get vitamin B12 deficiency. Okay, so next we will see about the clinical manifestations you will get in B12 deficiency. Now we will see about the clinical manifestations you will see in vitamin B12 deficiency. So we all know that vitamin B12 is required for relieving the folate trap. So if B12 is deficient, what will happen? The folic acid will be trapped as methyl tetrahydrofolate and it can lead to folic acid deficiency. So in B12 deficiency, you will be getting all the manifestations which you will see in folic acid deficiency, like megaloblastic anemia, elevated homocysteine levels, increased risk of cancer, etc. Okay, so like folic acid deficiency, here also you will be getting the similar features. Okay, in addition, as homocysteine cannot be converted to methionine, the transmethylation reactions will be grossly affected. Okay, so one of the important product we get from transmethylation reaction is choline, which is needed for the proper myelination of the nerve sheaths. Okay, so if this choline is not synthesized properly, it can lead to demyelination and neurological symptoms. Various neurological symptoms will be there. Okay, so this demyelination and neurological symptoms you will also get in folic acid deficiency, but in B12 it will be more severe. Why it is more severe means, apart from decreased choline production, there is also increased accumulation of methylmalonic acid. We saw that vitamin B, adenosyl B12, it acts as a coenzyme for our methylmalonyl CoA isomerase enzyme, which converts L-methylmalonyl CoA to succinyl CoA. Okay, so if B12 is deficient, that conversion will be blocked and it leads to the accumulation of methylmalonic acid. So this methylmalonic acid in turn, when it accumulates, it leads to abnormal lipid synthesis and it can also affect the myelination process. That's why the demyelination and neurological symptoms are more severe in B12 deficiency than compared to folic acid deficiency. Okay, so this demyelination, it can affect both the sensory pathways and also the motor neuron pathways. Okay, that's why this condition we call it as subacute combined degeneration. Okay, so along with folic acid, symptoms of folic acid deficiency, you will also be getting this important neurological manifestation in vitamin B12 deficiency. Okay, so you will be getting symptoms of folic deficiency and along with that you will be getting subacute combined degeneration. These are the major clinical manifestations you will get in vitamin B12. Okay, so next we will see how to diagnose this vitamin B12 deficiency. So how to diagnose it means, as folic acid deficiency is also part of B12, here also the lab investigations will also be similar to that of folic acid deficiency. That means what will happen? Serum levels will be normal. So what will happen means, in blood picture you will be getting macrocytic anemia. Increased MCV and macrocytic anemia will be the characteristic feature you will be seeing both in folic acid and B12 deficiency. Apart from that, in serum the homocysteine levels will be elevated. Then in urine, Figlu levels, okay, and ICAR levels will be increased in urine. Okay, so all these are actually part of folic acid deficiency. Okay, but along with these findings, one more important characteristic finding which actually differentiates B12 from folic acid deficiency is increased excretion of methylmalonic acid in urine. Okay, so which is otherwise called as methylmalonic aciduria. Okay, so that is one of the characteristic feature which differentiate B12 deficiency from folic acid. Okay, in folic acid you will be seeing all the other findings except this MMA. Okay, whereas in B12 along with this you will also be getting methylmalonic acid and sometimes even propionic aciduria also will be there. Okay, so these things when the patients are having the diagnostic features of folic acid deficiency along with methylmalonic acid and propionic acid in the urine, then you can make the diagnosis as B12. Okay, but it can give you just a clue, but for confirmation you have to measure the serum B12 levels. Okay, now we have lot of immunoassays which can directly measure the B12, which is actually present in very nanogram quantities in our blood. Okay, the normal serum B12 level is, it should be more than 150 nanogram per liter. Okay, if it is less than that, then you can make the diagnosis of vitamin B12 deficiency. Okay, so after confirming the deficiency, you should also identify what is the cause for that B12 deficiency. Okay, especially you should rule out the Addisonian pernicious anemia. Okay, intrinsic factor deficiency should be ruled out. Okay, if you are not ruling out, okay, once you confirm B12 deficiency, what you will do, you will supplement the patient with B12, oral B12 you will be giving. But if the patient is suffering from the intrinsic factor deficiency, even if you give B12, it may not be corrected. Okay, so you have to identify the cause, or otherwise you have to rule out whether he is having intrinsic factor deficiency or not. Okay, so how we can do that means by a famous test called as Schilling's test. Okay, so in this test what you will be doing is, first you will be injecting a patient with B12 injection. Okay, you will be injecting the patient with vitamin B12. Okay, so this vitamin B12 injection will have a good amount of dose which can saturate the storage reserve. Okay, the liver will be saturated after this vitamin B12 injection. Okay, so then after that you will be giving a oral B12 which will be having radioactive cobalt in it. First you will be giving injection B12. Okay, intramuscularly you will be giving so that the storage reserves will be saturated. Then oral B12 which has radioactive cobalt will be given next. Okay, so what will happen normally for a normal person, if the intrinsic factor is produced and all the other factors required for digestion are there, what will happen? This radioactive cobalt will be absorbed, okay, but it will not be stored in the liver because already we have saturated the liver. Okay, so it will be entering into our body and it will be coming out in the urine as the radioactivity will be cobalt will be coming out. So by measuring the radioactivity in urine, okay, if you get radioactivity in the urine, desired quantity amount if you get, intensity if you get, then it is normal. Okay, suppose if you are not at all getting any radioactivity in the urine, which indicates that the absorption is decreased. Okay, so it may be due to intrinsic factor deficiency. Okay, so in urine, if you are getting radioactivity, means it is normal, it is normally absorbed and it is coming out in the urine. If in urine, if you are not getting the radioactivity, means it may be due to intrinsic factor deficiency. Okay, that can be, it can be due to other factors also, but how to identify whether it is due to intrinsic factor means repeat the same test. Okay, now this time you do it with along with intrinsic factor. You give oral B12 with radioactivity cobalt, along with intrinsic factor you give. Okay, so with along with intrinsic factor, if the radioactivity is seen in urine, means the problem was intrinsic factor deficiency. Previously it was not there. Now when you give it, it is coming out, it is absorbed and it is coming out in the urine. Okay, so by that way we can confirm intrinsic factor deficiency. Okay, so this is how you can diagnose vitamin B12 deficiency. Okay, so regarding deficiency, you should know the causes, what are the manifestations and how to diagnose it. Okay, now finally moving on to the toxicity. B12 toxicity is quite rare because actually I said liver has a very good reserve capacity to store up to 1000 times of the normal dietary intake. Okay, so toxicity is quite rare, so nothing to mention about this. So to summarize, vitamin B12, we have 4 different forms: cyanocobalamin, hydroxycobalamin, methylcobalamin and adenosylcobalamin. In that, methylcobalamin and adenosylcobalamin are the active forms. Okay, so what are their biological actions? Adenosyl B12, it acts as a coenzyme for methylmalonyl CoA isomerase. Okay, so thus it helps in the catabolism of odd chain fatty acids, valine, isoleucine and methionine, which are actually the gluconeogenic substrates. Whereas methyl B12, it acts as a coenzyme for the enzyme homocysteine methyl transferase. Okay, through that it helps to relieve the folate trap, and also it helps in transmethylation reactions. Okay, now when this B12 is deficient, what will happen means folic acid will be trapped, so it can lead to folic acid deficiency. Along with that, as transmethylation is not happening properly, demyelination and subacute combined degeneration will also be getting. Okay, so how to diagnose it means they will be showing typical features of folic acid deficiency along with methylmalonic aciduria, which is the characteristic feature used to differentiate folic acid and B12 deficiency. That's it about B12. Thank you.